Okay, so good afternoon. I think that we can start on time. This is the third edition of our MDDB webinar series. I'm sure that you probably know about the Molecular Dynamics Data Bank project. This is an European Union-funded project that is trying to build the first European database on molecular dynamics simulations. The objectives of this project are clear, and you can see them in this slide. We are trying to apply FAIR principles on molecular dynamics simulations. That means that we are trying to move the biomolecular simulation community to the open science paradigm, something that is very important, especially in these years where the AI is revolutionizing everything. And the second objective is to try to integrate all of this information that we are generating with the molecular dynamics trajectories with the already available life science data ecosystem, starting with the PDB in Europe, the fantastic portal that you can find in the EBI EMBL group, which is a partner, as you can see on the right-hand side of the slide of MDDB project. Then we are also trying to define good practices, not only on the generation of the simulation, but also on the analysis, on the quality control, on the quality check of the trajectories. And finally, we are building the first prototype of a general repository of simulations, biomolecular simulations. And this is a distributed platform that we presented in the first webinar of this series. And you can find the video, the recording of this session in the MDDB website if you are interested. As I told you, all the partners from this European project, you can find them in the right-hand side, but this is not just a project that we are working on. This is a project that is mainly community-driven. That means that all these good practices that you can see here, the quality checking, all the metadata that we need to describe one simulation, all of this is driven basically by the community. So we are talking to the community, and the community is giving us clues, information, important points about what we need to store, what we need to analyze, what we need to share with everybody. And we have today a very good example of a community, which is the ABC community, the Ascona BDNA consortium that is working for many, many years now on simulations on DNA systems. And it's a pleasure, really a pleasure for me to introduce the recently appointed lecturer, Professor Sarah Hunter, tenure track assistant from the University of Barcelona, Dr. Federica Battistini. She's going to talk about the hexABC project, which is basically the last project of this ABC consortium that is trying to simulate a collection of 20 MER sequences, more than 300 simulations, more than 200 terabytes of data, a huge data set that is a fantastic example of the collection of molecular dynamics simulations that in MDDB we want to store and share with the community. So I'm now giving the floor to Dr. Battistini. Thanks for the presentation, and the floor is for you.
Thank you very much for the introduction. So,
sorry, I need to stop
no
sharing. Okay.
Okay. So, can you see the presentation correctly?
Yes.
Okay, perfect. So, hi, everyone. So today, as Adam introduced, I will talk about the hexABC project together with the MDDB. So first of all, I realized that in the title, there were two acronyms, so I would like to explain better what they mean. hexABC is part of a project of ABC Consortium and will involve a systematic characterization of all the unique hexanucleotide. Don't worry, I will go into detail during the presentation. And in this talk, I will show you how this is connected with the project, the European project MDDB, that is building a data bank for molecular dynamics simulation. So first of all, I will have to introduce, we can see that the common theme into the AB6 project and the MDDB are molecular dynamics. So molecular dynamics simulation have emerged as a powerful tool for studying DNA conformation. So during the past 3-4 decades, with the improvement of computational resources and with improvement on the force field, it was possible to use molecular dynamics simulation to analyze the structure and the flexibility of each possible sequence and also compare it directly to experimental data. At the atomic level and using, I'm explaining this now because later on in the presentation, you will see this picture over and over, analyzing the atomic structure, but also considering the base pair as bricks. So these two bricks, each base is a sort of brick, is able to analyze the movement of each bases along the DNA that will involve base movement and base pair movement, both rotational and translational. And here are all the movement that I will talk about, a few of them during this presentation. So as I say, molecular dynamics simulation was able to give us details at the atomic level, at the base pair movement, and also to give us ensemble properties for every sequence we would like to analyze and give us data that are able to be compared directly with experimental data. Considering this panorama, the ABC consortium, and I will do a bit of historical regression to show you and to explain you not only what they did, but why is important. So the Ascona BDNA consortium was founded in 2001 in Ascona and was a group of the most powerful groups in molecular dynamics simulation and the study of DNA. They decided to join the forces to produce a database with state-of-the-art MD simulation of DNA studying, as I show you in this square, a tetranucleotide, so the base pair and with the two neighboring bases. And they not only produce a data set for the molecular dynamic simulation of DNA, but also they try to develop standard and simulation protocols to improve and understand the base pair sequence and the effect of the structure of the dynamics. Along this year, as I say, I'm not going to show you directly the results, but why they are important. So in 2001, they started this project running 39 molecular dynamics of 14 MERS for 100 nanoseconds. At the time, it was a long time scale. And what they did was to characterize, these are the two main publications, was to characterize the 136 unique tetranucleotides, so all the conformation and the flexibility. And in particular, they center the point of view on one base pair that they saw that was very particular. There was the CPG steps. All this work, they shared the data with the scientific community and was very important, mainly for the development and the improvement of force fields to describe the DNA. So this work not only were very useful for the scientific community and experimentalists to show the structural DNA of every possible tetranucleotide, but also provide a data set to be able to improve the description of the backbone and the description of each sequence of tetranucleotide. Next step, in 2014, they used the sequences and basically with the improvement of the computational resources, they could and have a bit better ensemble of the conformational space with a longer time of molecular dynamics, so 1 microsecond. And these are the two main work that came out from that project. First of all, the mu-ABC that here, as I can show you, they describe the base pair parameters. Here we see the free translational one and the free rotational one between the bases. And what they have detected, being able to simulate for such a long time, is that some of the steps, here I'm showing you with the CG step, for example, they have a bimodal behavior. So they have two minima, or in this case, two maxima. So two structure They were the most favorite one, and this was depending on the neighboring step that the CG step had in the tetramer. Because, as I say, this was very important for the CG step, also for some biological function that the CG step has, like methylation or the intercalation. They focus more the attention on this base pair step, and they also not only analyze the base pair step, but also all the interaction with the backbone, the B1 and B2 form, the presence of the ions. So they went deeper and deeper in studying this base pair. As I said before, all these datasets were available to the scientific community, and they helped a lot for the further improvement of the force field. So from that, they came out to two of the main force fields that are used nowadays for the DNA simulation, PAR-BCC1 and OL15. All this data and all the improvement and the data that were collected for MDDB were the stone that they build on top all this improvement for the force field. The furthest step sorry, was the mini-ABC work that was done in 5 years later in 2019, a 1 microsecond simulation of 52 molecular dynamics of 18 MERS, so longer, a higher amount of simulation and for longer sequence. They allowed the study to analyze every possible unique tetranucleotide for a long time scale and analyze all the bimodality. Don't worry about these plots, later on I will explain better. All the bimodality, also all the possible behavior in the backbone moment, the correlation with the base pair, and the different force field. This is a set that is right now, the most updated one for the base pair parameters was used through this year to develop the coarse grain model. So all this data on the atomic level for every possible nucleotide was very important to build a coarse grain model to analyze the correlation in motion of the DNA and to feed like the features for many machine learning approaches to study nucleosome formation, transcription factor binding. So they were important, all this data for AI that is the new development in the most developed software in the past years. This data were available not only for the partners, but also to the community. And they built one of the first database for molecular dynamics simulation, not SQL database, where they downloaded, it was possible to download the trajectory, but was like a sort of small, because we can see the amount of data, database for the molecular dynamics simulation. And now I will go through what we are doing right now, what we are collecting right now. On the right, like Adam say, we are the partners of the ABC consortium right now. We have 13 groups all over the world that are collecting molecular dynamic simulation to be able to study, to pass from the tetranucleotide to the hexanucleotide. So all the possible environment of in the hexamer, at the hexamer level of the central base pair of DNA. In this work, we didn't only extend the hexamer, but with the usage of all this community, of the power, computational power of this community, we were able to extend also the time length of the simulation and get closer and closer to the experimental timing. So be able to see, and I will show you in this presentation, some behavior that have been seen experimentally, but not yet in all the molecular dynamics simulation. So this is called the hexABC. As I say, we are analyzing all the 2080 hexanucleotide. And I am showing now basically the step that we follow, the protocol that we follow in this project. So first of all, we build the model structure, we generate the topology, build the box water with OPC water molecule, add the ions, and we started the calibration with minimization, MVT, MPT calibration, and we follow all the protocol. That was the newest one available, the most updated one and the most refined one that was Daniel Riro and Bernard Brooks' work. As I said before, the ABC consortium was founded not only to build database with all the properties of DNA, but also to show to the community the best practice for DNA simulation at the time. So for us is nowadays. So this project is divided into two main parts. One is to define the best practice for DNA simulation, and the other one is to collect all the data. In this talk, I will focus more on the second part, but the first part is basically following all this protocol that obviously have been tested, compared with all the possible force field and conditions that nowadays are available. So what we collected for each sequence that we simulated are 4 microseconds with a time step of 4 femtosecond, considering using hydrogen mass repartitioning. We collected the data every 20 picosecond, and being a big community with many partners all around the world, we had to define a very established protocol. So every dynamics is divided into 200 chunks of 50 nanoseconds. So every sequence is composed of the same chunks, and every simulation is coherent with all this protocol. Afterwards, we analyze this simulation, and I will show you later on using also some workflow of the bioBB project. So now we'll go a bit more in detail of the hexABC project. So we pass, as I say, from dimer to tetramer to hexamer. So we analyze every possible unique hexamer with the central tetramer, changing the central tetramer and the basis at the end. This ended up in being able to simulate 190 sequences that are 20mers with the CG at the extreme, not to make suffer the central part from the terminal effects. And we divided them, the simulation, in among all the partners. This ended up in 100 simulation of 20mers. We ran 2 replica. We ran more replica for some cases that we were a bit in doubt if there had some problems. I will show you later some example. We collected 10 microseconds for simulation for a total of 162 terabytes, so around 200 terabytes. All the simulation were divided among the different partners, and we had 60,000 trajectory chunks, so more than 500,000 files. You can imagine the amount of data and the problem with the transferring the data and organizing everything in a very consistent way. Fun fact that is not that is real, like all the previous projects, so ABC, MuABC, and MinABC, before sharing the data with the community, between the partners, we were sharing the simulation, and we were downloading the simulation using disk, like using mail, and sharing our simulation through the post. But right now, we have a newest technology, so we were able to share the data in a faster and more reliable way. So first of all, thanks to the Barcelona Supercomputing Center and the program of the Red Español de Supercomputación, we got a project that we gain a storage of the data in the Barcelona Supercomputing Center for 5 years of 1 petabytes. After that, we collected all the simulation from the different partners, and we transfer all the data to the BSC. We did it from a more traditional way with the virtual machine, but also using the newest technology like Globos that gave us a safer and faster transfer of all the data. I remind you that we are talking about around 200 teras of data. And also, like to be sure that we had a backup copy of all the data we are storing in the BSC, we use the fast secure transfer and reliable of Globus back to another node that is in the University of Utah, where we have a database. We have the backup copy of the database in their cluster. in all of this transfer of the data, we had to be sure also that the data were consistent, as I say, and we didn't skip any frame, and the trajectory were like following, we didn't skip any frame. This was a very careful job that we did. So, and it's something that sometimes we have the simulation, the molecular dynamics simulation, and we don't go into detail of each frame. We did also to be able to see if we had a problem in the transfer of the data or in the production of the data. And we saw that sometimes we had corrupted frame, or sometimes, for example, as you can see here, when there is not a distance between the different chunks of 20 picosecond, that is our time step, we had sometimes an overload, writing of the data one on top of the other, or sometimes, as you can see here in the 40, we were skipping one of the base pair. So once we detected one of these problems, we rerun the simulation, and so to be sure that every simulation that we are running is consistent with the same time step. As I said before, we did the setup, equilibration, MD production, and transfer of the data, and checking up of the trajectory. And then we run through the analysis of each trajectory. I will show you I will show you now some of the analyses that we are running, just one of the most typical one to check the stability of the DNA, and some more fancy analysis that we are doing to see the influence of the hexamer level. And also very important is that all the partners that are in the community are able to access to the trajectory, not only to store and to transfer the trajectory, but also to be able to run the analysis. And they can connect directly to our cluster in the Barcelona supercomputer. As I say, now we'll show you, some of the analysis that we are doing. So first of all, what we did was to analyze the stability of each trajectory, running a root mean square deviation starting from the starting of the conformation, and we checked that the RMSD was low and the trajectory was stable. We ran also the RMSD, and we also compare each sequence with the fiber BDNA, with the ideal BDNA, and this is the distribution of the RMSD for all the 380 trajectory for the first 200 nanoseconds, so we can see a very low RMSD and a stable behavior of the starting of the trajectory. And we check also the last part of the RMSD. So the last 200 nanoseconds were useful to see, first of all, the trajectory had homogeneous and very stable behavior around 2.5 angstrom, but also was very important to detect the trajectory that had a higher MSD and to check what was going on. I will show you later two examples. For the trajectory that we have seen that had a weird behavior in one of the two replica, we ran other replica, 3 or 4, or we extended the time to be able to see if it was a singular case or was something that was systematic given by the sequence. Then what we did was to analyze all the pairing of the base pair, and what we analyzed was the stability of the hydrogen bond. So here is one example for one of the sequence that is on the x-axis along the trajectory. We check if along the trajectory we were maintaining 3 hydrogen bond in yellow, 2 hydrogen bond like the AT base pair in green, or if we were losing hydrogen bonds, so having one with the dark blue or with the light blue, or we were losing the base pair. So as you can see here, we have a bit of opening in the terminal base. This is called fraying, and can be given by the opening of the terminal bases. So this, we ran it, as I say, for all the trajectory, and on average, we had this stable behavior. But for some of the trajectory, we detected some wide opening, like I show you here. This is one of the trajectories that show a higher MSD in the distribution. And if you can see here, we saw a wide opening of the DNA that is something that experimentally have been detected a few times. But as you can see in the video on the right and also in the plot here on the right, after 100, 200 or 300 nanoseconds, this DNA is able to recover the DNA conformation. So as I say, in this project, we are running a lot of molecular dynamics, but also in a time range that is very long compared to the standard right now. So we can see behavior like not only the opening, but also the recover of the double helix of the DNA. This is just to show you that this one was one of the replica, but when we ran other 2 or 3 replica, this behavior was only in one of the case. Also experimentally, we have a percentage of when it happens, the frame, it doesn't happen always. So this is very consistent with what have been seen experimentally. Also, we saw something that is very interesting. So if you're not an expert on DNA, if you follow me here at the bottom, usually when we call the base pair parameter, the base pairing, we are talking about the Watson and Crick. And you can see here the hydrogen bond pairing between the thymine and the adenine. This is not the only pairing that it can be seen in the DNA. We can also have pairing that is called oxine, when, if you can see here the adenine, we have a shift from the antigen to the syn conformation, and here we have a different pairing with different hydrogen donor and hydrogen acceptor. This behavior has been seen in NMR, in many experiments, and in some of the molecular dynamic simulation, and but we didn't have like an till now an ensemble of this behavior, and also that was comparable to the percentage that have been seen experimentally. In this project, we did, and here I show you like this pairing that is here in the light blue. Now we are in the Watson and Crick, and you can focus on this adenine. You can see that is flipping from the anti to the syn conformation, and is now forming the Watson here, from the Watson, if you can see here, to the oxine pairing. This, as I say, is very important because it can be directly connected to experimental data, and also allow us to see biological behavior that before has been seen, but only in just a few of the cases. Here, as I showed you before, this was one of the examples of the replica, but replica 2 and replica 3, we didn't see this behavior, and our percentage of forming the oxine is very consistent with NMR data for this sequence, for this environment. We check, as I say before, also base pair parameters, so the different structure and the different flexibility here, the first concept of the movement between in the base pair step, a translational and rotational one, shift, slide, and rise for the translational one, and tilt, roll, and twist for the rotational one. We check the average parameter for each of the base pair in the sequence with the standard deviation and the groove width and depth. All these values are very consistent with the data of NMR and X-ray that are are stored in a database for every possible sequence. So we could reproduce very well all the experimental data. And now I want to show you something that experimentally is a bit more difficult to detect. Sometimes it's possible, but not always. But we could study it in detail using the molecular dynamics simulation. So as I showed you before very briefly in the MDDB project, what they detected is for, here we have the CG base pair, for example, with a different environment. Here are the tetradenucleotides, so the base that are the neighboring bases of this dimer. What they have detected is that for some parameter, for example, slide and twist, some bases, they have a unimodal behavior that can be on the average, lower or higher. So you can see here there is a Gaussian distribution. For some parameters, they could detect a bimodal distribution. So there are two possible conformations that the same base pair with the same environment, they could sample. So as you can see here, we have the same dimer CT that, depending on the neighboring step, could have two minima, one, so being unimodal, or have other two minima that are completely different. The same for twist. I show you here 3 examples. Here we have the bimodality, so two minima for the twist parameter, so this rotation. And here for the same dimer in a different tetrameric environment, we have the unimodal. So at this step, and was part of the work that they did in MUABC, we are considering the dimer in a tetrameric environment. Now, as I say, we are extending to the hexameric environment. So this is what we get. Don't worry, I'm not going into details of each of these line and plot. I will show you just one example. But here what we did was to analyze for every possible movement the different tetramer in a different hexameric environment, and this one on the y-axis. Here are 2 examples. I'm showing you 2 of the parameters that are bimodal, slide and twist. And here you can see for here on this part on the left part of the of my screen, you can see the tetramer ATCG with, with different neighboring step, X and Y, to generalize. You can see here that for some of the parameter, we have bimodality, so there are 2 minima, and then changing the neighboring step, X and Y, we have a unimodal behavior. So what they've been detected that is at the tetramer level, that depending on the neighboring step, we have a unimodal or bimodal behavior. This doesn't depend only on the tetramer, but has to be extended also on the hexameric environment. And this is very important because it will allow us to have more and more information and a more detailed information about the environment. On the right, another example, but in this case, we have 3 minima. So we can actually consider 3 possible clusters of conformation. And for the same base pair, for the same tetramer in different hexameric environment, sometimes we have a bimodal behavior, and sometimes we have a more accentuated unimodal behavior, like here on the top right. We have collected all the data, all the possible minima, and from here also all the flexibility of each base pair. This can be used, as I say, for experimentalists to check the data, or for other study at the structural level and choose the best sequence then for some experimental or computational work. Here I'm showing you 2 of the new analysis that we are running. This is an ongoing work. What we also check is what is called frustration. So what we are calculating right now is the harmonic energy, because we collected, as I showed you before, all the minima with all the flexibility, is considering the same base pair, for example, CG, changing 1 or 2 or all 4 letters, so the neighboring step, to see how much is the energy to go from this minima to the minima of the new energy. So what we have seen, and here I show you like a zoom in of this big matrix for all the central dimer CG. We have some environment that are more frustrated, so we will need a lot of energy to pass from one hexamer to the other conformationally, and some other that don't influence much. So it looks like that also energetically, the neighboring step of one dimer is influencing one conformation or the other. But at this step, we are checking only how to pass from one hexamer to the other. And what about along the sequence? So is one state in, for example, the first tetramer influencing the conformation of the next one, and next one? So this is what we are considering is the correlation of the states. So here at the bottom, I'm trying to show you some examples. So for example, here we have a base pair at the step 2, for example, with 3 minima, and we have some steps that are in this minima, for example. Are they gonna be represented, are they gonna be represented in the next one, the same high twist, for example, high twist, or are they gonna be moved in the low twist? So are the neighboring step correlated, also when they go from a cluster to one cluster. So what we are studying right now, sorry, there should be a video, okay, are for each tetramer how the different base pair are moving and all the cluster of the conformation are moving from one step to the another. And we are looking if these correlation are random or they are directly correlated through the parameters. As I show you right now is an ongoing work, as I say, of all the data set and all the analysis of the DNA structure. We have 190 sequence of 20 mer of DNA structure, and then we have the 2 replicas. So this involved, as I say before, 190 molecular dynamic simulation with a collection of 200 terabytes. And now we are in the phase of having all the analysis with quality control, helical parameter, principal component analysis, correlation, and many others. All of these have been, all the analysis and all the simulation, and in particular the analysis, can be stored in a no SQL storage, MongoDB, that contains the MD coordinates, analysis, and all the metadata. And all of them can visualize and then analyze access. So as you can imagine, at this stage, we were like, okay, we have many data, and soon we would like to share all this data, not only with the ABC consortium, but also with all the scientific community all around the world. That's why we talked with the project, the people that is leading the project, the MDDB, that they are building a European repository for biosimulation data. So MDDB stands for Molecular Dynamics Data Bank, and for them and for us, the hexABC was a good use case because it's considering one of the one biomolecule, so DNA, and we have many data that we would like to share. Not only data, but also analysis, because as we want to establish with the community our data, and we want to follow the fair philosophy, so to have all the data that are findable, accessible, interoperable, and reusable. So we contacted the MDDB, so we are in touch with the MDDB project. We gave the sequence, and the importance of this project is that not only gave us a platform, a repository where to store the molecular dynamics simulation, but also in this project, they built a web-based GUI. So they have NoSQL and a graphical user interface where you could be able to see the trajectory and see also analysis on the trajectory, some of the analysis that I showed you before. So we have, for example, principal component analysis data, and here at the bottom are the hydrogen bond analysis that as I showed you before, but the power of this web-based repository is that, for example, if we look at one of the base pair, clicking just on one of the frame, the user is able to see directly the hydrogen bond that is forming, and also he can then go through the trajectory and monitor all these hydrogen bond. That for us was very good because we can monitor the oxygen, for example, base pair formation, how the flipping of the base from the anti to the syn conformation, the exposition of the base, the fraying. So we're able to monitor everything in just looking at the at this easy web tool. And also they can analyze, for example, the pairwise RMSD to be able to detect, for example, when some frame is skipping, when we have a high RMSD, so something happened in the trajectory, and to see if it was part of maybe problem in the movement of the data and transfer of the data in the production, or something that is really happening to the molecular dynamic simulation. So everything has been checked, and everything is checked and is easy to visualize. Another powerful thing of this MDDB database is that there is not only web-based, so it's not only able to visualize, but there is also a programmatic access. So you can imagine that, for example, in this project, we have a big simulation with a lot of data, but maybe one user is just interested in one part of the simulation, maybe the last part of one of the sequences, because maybe they're running experiment through the sequence, so they want to compare their data, or maybe they're just interested in one part of the sequence, so the central part or the terminal part. And with this programmatic access, it's possible to not only select the sequence, but select, for example, the frame that the user is interested with, or the part of the sequence that can be downloadable. So it's very user-friendly. This is basically this philosophy that is followed in not only in the hexABC project, but also in the MDDB, and is what we share with them, and that's why we join forces and we give them the data. So once we have the data, the three steps that are the most important is not only to have all the data of the simulation, but also to report them. For example, there is one part of the metadata, so it's possible to see which one is the sequence, where it's being run, and all the detail on the trajectory that the user is visualizing or downloading. And then they have an analysis support. So here, for example, I show you the electrostatic surface that maybe is very important if someone is interested to check maybe when a ligand could bind to the DNA. And but not only keeping everything like data reporting and analysis, but also the action to be able to download the analysis, download the data, because we think that is very important, and mainly for this big project, to be able to share the data with the scientific community, because as I showed you before, previous projects, the data set that was built with the ABC was then used for coarse-grained model, for feature for AI. And I think that I'm sure that this data will be used later on for improving, for example, force field, and to have better and better result in the next years. So I would like to thank, first of all, the group I'm working right now with, and the MDDB project, SICAM, Barcelona Computing Supercomputing Center, Modesto Orozco, that is the PI of this project here in Barcelona, and also the ABC community. Here is a picture that we took like almost 2 years ago in Ascona, where there was the reunion, where all the groups joined together to establish all the path of this project they were doing, and possibly the next one that we will have in the next year. So thank you for giving me the time for this presentation, and any question, comments?
Thank you very much, Dr. Battistini, for this fantastic example of a project that on the one hand is a huge scientific project that you have shown us what you can extract from all of this information, extending from the tetramer level to the hexamer level, and all the different values of, for example, the helical parameters when you extend from the tetramer to the hexamer. And on the other hand, also a nice example, a perfect example, I would say, for us, for MDDB, for the importance of storing and sharing this huge data set of information. And your examples at the beginning of the presentation, I think that are fantastic for this. So this data and the possibility to reuse and reanalyze all this data for the scientific community is also giving the possibility to for different projects and different research projects in science. I'm not seeing any query and any query, yes, in the query and answer section. So I'm gonna give you a few minutes to think about questions and write the questions in the query and answer. at the bottom of your Zoom panel. And in the meanwhile, I also wanted to thank Dr. Battistini for being perfectly on time. So we have 15 minutes for queries now. And I have one if, so that giving a few minutes for the participants here to think about different questions. I have one that is, I think that this is a really huge project. So many partners involved, worldwide, collaborative project. I'm sure that this hasn't been something easy. So it's been many years on the project. So can you give us some ideas or suggestions on what is the most complicated part on all this huge project about generating data, storing data, analyzing this huge amount of data, and some suggestions about these high throughput projects for us?
Let's say that this project came after previous projects. So we're not the first one where we're collaborating with these groups. So what we did, I think at the starting point, and was one of the good steps, was to be able to build a protocol, as I say, very strict protocol, to be able to have all the trajectory with the same chunks, same protocol, same input, same output. So this gave us an easier way later on in the checking of the trajectory. One of the most difficult step, I think, that was the transfer of the data, but the new technology like Globus was able for us, and the storage of the BSC gave us a lot of help to be able to transfer the data. So what I think that was the hardest part was to check that all the chunks were there, all the trajectory, and there were no problem. And to be able to detect if the problem was in the production of the molecular dynamics or in the transfer of the molecular dynamics. Apart from this, then when we collected all the data, then is to share all the data for partners and community. But for partners was a bit easier for us because they had access to our cluster. For the community, we are in the hand of the MDDB right now. So I hope that I answered to your question.
Yes, thank you about this. We have a couple of questions now in the Zoom section, query and answer section. The first one is from Charlie. He says, "Hello, Fede, great talk. One question, this concept of frustration seems very interesting. Where do you think we might see this in the biology of DNA or maybe DNA protein recognition?"
I have been analyzing this data in this past week, so it's very fresh. What I think that we will see are in the base pair that are very bimodal. So we know that, for example, CpG step is very bimodal. That's why, for example, in some biological cases, it's been methylated, or there are intercalators or protein that recognize only the CpG step. And I think that changing the hexamer environment or the tetramer environment and being able to define this energy to move from one step to another will give us a bit of light or explanation why some protein binds to the same base pair or the same intercalator, but with different hexameric environment. And this has been seen biologically, but we didn't know why this definition of the extreme part, but I think that this different in the structure is gonna be a huge step to understand some biological behavior.
Thank you. More questions. One from Oliver Henrich. I might have missed it, but was wondering how big the quantitative difference in bimodality is between considering tetramer and hexamer contact.
I mean, I didn't show you like quantitatively how much it happens, but this is basically what I seen in the maps, and we are collecting all the data. But there are many cases of steps that are bimodal that in some hexameric environment they are not. So it's a bit. Like, I don't know in percentage, I don't know quantitatively, but I think that they will make the difference for many of the hexamers. So we have seen it repeatedly. So it's not just few cases.
Thank you.
I mean, soon we will have all the data, so we will publish then and you will have more.
So we have another one from Chantal Prevost. Thank you very much for this very interesting presentation and for everybody participating to this work. Thanks, Chantal, for this. I was wondering whether hexanucleotides would be enough to observe collaborative transitions like what happens upon DNA binding to architectural proteins, TVP being the most extreme case. This involves opening the minor groove and unwinding over at least 4 to 6 consecutive base pairs.
What we're seeing right now, for example, I've seen many of the project for DNA protein binding. So a 6 is quite of a nice length, like is quite of a length that is enough to describe, for example, the minor groove or the major groove. Obviously, we are also checking if there can be an octamer effect. Until now, for all the data we are collecting with duplicates, we don't see the same as we are seeing for the hexamer, that the octameric environment is making a difference. So will be only like to connect a different hexamer and see if we could predict, and will be very interesting in the future to see how all this data could be effective to describe the binding of architectural proteins, and if we will not be enough to extend. So maybe we will have another ABC project later on. But for now, I'm seeing that for transcription factor, for example, and for the description of the grooves, hexamer are quite good length, and octameric is not influencing much the structure.
Okay, thanks. So we don't have more questions. And with this, I would like to thank again our speaker today for the presentation and also for answering all these questions. And we will finish the webinar with the presentation of our upcoming MDDB events.
Thank you for the presentation and the invitation.
So you have here in the screen that the upcoming events, we have a couple of new webinars from our webinar series, one in March and one in May. The first one related to membrane proteins from the memprotmd database, the second one for protein-protein interaction systems with the DynaRepo project. And finally, the announcement of a new MDDB workshop in Lausanne. When data science meets molecular dynamics, the registrations are already open in the CCAM website. The program will be soon available, so take a look because it's going to be really interesting. And with this, I'm closing the session. Thank you very much for all the participants in this webinar. Thanks again for the speaker, and hope to see you in the next webinar for MDDB.